All right, thank you for joining us today for the Kirby Institute seminar series. My name is Rowena Bull. I'm part of the VISP program at the Kirby Institute. And I would first like to start by acknowledging the traditional custodians of the lands of which we are gathering today and pay my respects to the Bedical people of the Eora Nation. And I would also like to welcome any past and present Indigenous members that are joining us today. So just some housekeeping before we begin. The format for today will first be a seminar, and then this will be followed by a Q&A session afterwards. If you're joining us online, there is a Q&A icon down the bottom of the screen where you can start to enter your questions as the talk is proceeding, and we can address those towards the end of the seminar. And for those of you that are in the room, there's some microphones that I'd like to ask you to use so the people online can also hear. Okay, so without further ado, we will begin the seminar. So today we have Dr. Alexander Underwood is joining us. He completed his PhD at the Kirby Institute in 2020, and he then went on and obtained a prestigious Bridge Fellowship in Copenhagen with Jens Buch, who is a very eminent HCV virologist working over there. And he initially went to start working on hepatitis C, but in 2020, a lot happened. And like many of us, he moved more in towards working on SARS-CoV-2. And today he's going to take us through some of that work that he'll be looking at. Thank you.
Thank you for the introduction, and thank you for organising this. Firstly, I would like to acknowledge the Bedical people of the Eora Nation and pay my respects to elders past and present. So a great deal of my work in Copenhagen is on neutralizing antibodies. So I thought I'd just present everything I've done on neutralizing antibodies. And I know it's post-lunch, so I'll try and keep it as short as possible. To start with, I thought I'd give a brief introduction to who I am. So I did a Bachelor of Science with Professor Hazel Mitchell at UNSW in BABS, and then did a PhD with Roe for several years after that. And then I gave a presentation. I found the event actually online back when I did my PhD, and that was like my final get-together of everything. And I'm currently doing a postdoc with Jens Buch, as Roe mentioned, and supported by a Bridge Fellowship until 2024 and 2025. Let's see what happens. Hopefully moving on. Anyway, a lot of people ask me why I moved to Denmark, particularly Danes, because they are so fond of Australia. They have no clue as why an Australian would come to Denmark when it's a bit colder, a bit more grey. And it sort of dates back to when I met Jens at ACH Squared in 2018. We kind of got on well there. And then later in the year, I went to Dublin to the HCV conference, and we met again and got chatting. And then the following year, I went to the HCV conference again in South Korea and gave a nice presentation. At the end of the presentation, I kind of sold myself and said, I'm looking for a job. Has anyone got anything? And then Jens approached me and said, would you like to come to Copenhagen? And the rest is kind of history there. And I put up a couple of photos of the more touristy area of Copenhagen that everyone loves. And this is the view we get from the mast tower, which is one of the places I work. And this is a photo of me at the start of the pandemic when we moved to the pandemic research. And it was really nice going to Copenhagen because we had a couple of ISPAs over there as well. So that was, I made a nice and easy move. So I thought I'd introduce the CO-HEP group to start with as well. So we have Jens, who's the head. He did his postdoc in NIH in America and then came back to Copenhagen and set up a team with Professor Nina Weiss, who is sort of the clinical head who works at the hospital and has a whole clinical team there. And then under Jens, we have Judith, Trolls, Yannick, and Sansa, and they all have their different areas of research in virology, immunology, and all sorts of bits and pieces, and vaccine development as well for hepatitis C. And although it's a Copenhagen hepatitis program, we've now branched out to respiratory viruses as well, such as coronavirus. I actually got this slide from Jens himself, and it's basically just to show the amount of members that we have as part of CO-HEP, ranging from professors to all sorts of different types of students and all the different nations we have with the flags down here, which is really nice. So I thought I'd start at the beginning when I first moved over, and as soon as the pandemic happened, we got the lockdown, and I get a call from Jens saying, we're not going to be doing any pandemic research, and nothing to do with coronavirus. Everyone's jumping on the bandwagon. We won't be doing that. 2 weeks later, I get a call saying, guess what? We're jumping on the bandwagon. You're coming back to the lab. And we started setting up a cohort called the Clinical, Virological, and Immunological COVID-19, or CVIC for short. And this was initially just whoever we could get. So mild infected, non-hospitalized individuals. Then we got hospitalized individuals. The vaccines came out. And we had both homologous and heterologous vaccinations, and I'll go through a bit more about that later. And to start with, we did T-cell epitope discovery, which failed miserably. And then we moved to neutralizing antibody response, which is more of a safer bet, and the work I'll be presenting today as well. And more of what I'm doing now is characterizing antigen-specific T cells, also B cells with 10x genomics and more fancy techniques, which is nice. I'm not going to go into too much about SARS-CoV-2 because everyone knows everything about it, but basically more than 600 million infections to date, more than 6 million deaths, causes various disease states from asymptomatic to severe, is pretty genetically diverse now, or I would say relatively genetically diverse, with many classified variants, as I'm sure you're all aware as well. The spike protein is the major target for neutralizing antibodies. We have a series of different vaccines available, with the newer ones being this bivalent vaccination. And the only important point I'd like to address here is that in Denmark, and I think Norway as well, AstraZeneca was put on hold in March 2020 because of this thrombotic issues. And then afterwards, people got an mRNA booster, whether that be Pfizer or Moderna. And what that meant was we had these heterologous vaccinations rather than the sort of homologous Pfizer, AstraZeneca, or whatever. So to start with, what we wanted to do was establish assays using whole virus as opposed to pseudo-typed models or surrogate models. We published quite a nice paper, basically taking inoculum, putting it into viroESIC cells, passaging it and growing up like many other labs. And we didn't have a PC3 facility, which we kind of needed. We got a PC2 plus, which is just kind of accepted for SARS-CoV-2 work with all the apparatus on and everything like that. And essentially all the first author did here, Sansa, was take the stock virus, which had a couple of mutations. Can I minimize this? Yep, great. A couple of mutations to begin with, which was the classic D614G, which everyone knows about, and another sort of less prevalent mutation, this E309K. Grew it in Hu 7.5 cells because they were kind of the only other cell line we had around, but it didn't grow very well initially, but then became adapted. And the nice thing was that adapted virus grew a lot better in all these different cell lines, including lung epithelial cell lines, CALI1 and A549, showing the original here and then the adapted in infective hepatitis here. And then what we found was there was several substitutions and a deletion. This is quite a common site for a substitution, this 484D, but there was a couple of other ones which are related to different sites, cleavage site, for example. And what I did together with my PhD student, we looked at recapitulating this data in the pseudo-type model where you only have the spike protein, don't have any of the other proteins there. You use some other backbone. In this case, we used MLV Gagpol, again, because we already had it available. And essentially, all I want to show is we looked at the single mutations. We also looked at some combinations, but essentially what we found was you needed the full variant or the adapted variants, the whole spike, all the mutations, to have this sort of adapted infectivity to all these different cell lines. And what was nice was we could then infect these cell lines, whereas previously we couldn't in the pseudotype model. And so from here, we now had a virus we could use. We could use the adapted variant. We could use the original isolate, and we wanted to test neutralization, which was kind of what everyone was doing at the start of the pandemic as well. And so we collected a lot of plasma, and then the assay was essentially trying to be as simple as possible, do twofold serodilutions with virus at a specific MOI, which could infect a single well without causing cytopathic effect. And that was our kind of standard for all different viruses as well. Infect viroESIC cells as a primary cell source, 48 hours incubation, and then we would do immunostaining using DAB, which basically would stain for the spike protein, and you get this brown precipitate, which we can then count on a CTL machine, and you get your FFUs, and you compare to the control, and you can get your neutralization as so. And then based on the serodilution, you can calculate your inhibitory dilution, or your 50% inhibitory dilution, or ID50, which is what we used pretty much from now on. So back to the paper quickly, we compared the original wild type, I guess you could say, isolate versus the adapted isolate, and there was no real differences in neutralization. And then this was also recapitulated in the pseudotype model. The only difference was that the pseudotype model required higher dilutions to neutralize it. Not entirely sure why, or if anyone has or sees the same thing, but that's what we found. But it was just nice to see that we could use either one, and we could also use different cell lines to do neutralization if we want to as well, such as more appropriate cell lines like the lung epithelial. So going back to the cohort, it was an evolving process through the pandemic. It was kind of like whoever can join, we would like you to join, please, and we'll take your blood, PBMCs, and have a look at it over time. And like I said, to start with, we had this non-hospitalized group, and we just got them whenever we could post-infection. So it's kind of ranged from half a month, sort of 2 weeks, to nearly 6 months. And then we would take follow-up samples at 6 months, 12 months, 24 months, and try and be a bit more organized following the samples. We then got hospitalized subjects, and because they were hospitalized from the infection, they was much closer to the infection dates, and we had a lot more samples because they were in hospital, a lot easier to take. We didn't need them to come in and take samples that way. So that we had a lot better follow-up here. We actually had a 24-month follow-up now as well. And lastly, we have these sort of the vaccinations. So we have the mRNA, which is the homologous Pfizer in this case. And we had vector mRNA, which was AstraZeneca and either Pfizer or Moderna. And this was a bit more all over the place in terms of sample follow-up because people got vaccinations at different times, particularly in the vector mRNA group, because there was a hold on when you were going to get the booster. It was a bit ambiguous when we're going to get these follow-up samples, but we did our best. And then basically these syringes here indicate the possibility of getting vaccinations, and the viruses here indicate the sort of window in which that variant was prevalent and an infection could occur. Yep. Yeah. Just the first two. Yeah. Because just the two. So prime boost. And then the third one, yeah. So we have yeah, exactly. In this case, but the vector mRNA was AstraZeneca, Pfizer, or Moderna. No. One prime, AstraZeneca. Then, yeah, boosted with mRNA, Moderna or Pfizer. And then the third shot was Moderna or Pfizer as well. Because AstraZeneca got scrapped. Because of the thrombotic issues. They were too worried about that. Yeah. Yeah. It gets a bit more complicated, yeah. Because then there's also infections in between. But yeah, trying to classify the groups becomes complicated as the time goes on, yeah. But essentially, yeah, so this refers to only the prime, the first one, and the second one. But the booster was always an mRNA because yes, any vectors got scrapped, including the Johnson & Johnson one as well. And we also have now, which I'll talk about later, the bivalent booster, which is very new data, but I'll present it anyway. So just going to talk about a couple of papers we published quite early on, which was nice. Essentially, in summary, looked at these non-hospitalized, very mild COVID individuals. And we looked basically at baseline to 6 months and how the neutralizing titers looked. And so what we found was generally they were quite sustained. And the closer you took it to the 6 month mark, the baseline sample, sorry, the less difference there was. So essentially what we found was that the first month you get the peak response and it dies off quite a lot and then sort of sustains out, which is a pretty standard response in acute responses, I think. Not particularly exciting, but it was a result, which is kind of nice. We then followed up with the same sort of analysis, but on the vaccine samples. So first thing we want to look at was prime boost. And clearly you can see there's a nice boost from the vector to the mRNA. Then we looked at 1 month post-boost for both groups. So the mRNA, vector mRNA. And you can see they generate very nice neutralizing antibody titers. But interestingly, the vector mRNA group, so the AstraZeneca, Pfizer, or Moderna group, actually had higher compared to the Pfizer, which has been published by others as well. So, but nonetheless, an interesting result. Smaller sample size, albeit, but kind of nice to see. And we compared that to time-matched mild COVID-19, so 1 month post-symptom onset, and we found that the vaccination was superior. We then looked at the vector prime only and compared that to the time-matched mild COVID-19, and you can see that actually the infection was superior to the prime vaccination only. So you need that prime boost to then be better than the infection, at least for mild COVID-19 or a non-hospitalized infection. Against ancestral strain. Yeah, so we didn't have any of the other isolates at this stage. It was just all against ancestral strain. And yeah, from this paper now, then we started getting all these other isolates, and I'll go into that, like more a breadth analysis as well. And shortly after, which was really nice, and you'll see probably, I'm sure everyone's read these papers as local papers, so it's really nice, that neutralizing antibody responses correlate well with protection, I believe, from disease. And it differs between variants as well. And again, very well, nicely published papers. And it was great to see these these names out and about as well. So it sort of established why we look at neutralizing antibodies as they are a correlated protection. And more recently, so a couple of months ago, published another paper, again on neutralizing antibodies, but this was the whole cohort now. So we've included as many subjects as I could and just screen everything, ranging from neutralizing titers to the ancestral isolate and all these other isolates as well. The first thing I wanted to characterize and sort of eliminate, though, is re-exposure or exposure, because obviously that's another antigen exposure that can change your whole profile or antibody response. So, of course, we know that in the vaccinations, particularly these ones, there was only the spike protein used. So antibodies targeted against the nucleocapsid protein, which should not be there. If they are there, then that means they've probably been exposed or infected. So we developed an nucleocapsid ELISA that basically just screens for antibodies to the nucleocapsid protein. And so it's easier to show the bottom two because that's the vaccinated group, and you can see that there's huge spikes following whatever time frame. And some of these we could easily correlate with PCR results as well, which is nice. But basically, we can now eliminate these as infections and additional antigen exposures. And similarly for the infected group, what we should see is in a boost in the nucleocapsid response. We define that as a twofold boost, basically, as a re-exposure. Then looked at the durability. So how do the antibodies look over time? And you can see there's subjects are taken at all these different time points. And the idea was to put it as days post-symptom onset and kind of characterize them and time match them as best as possible. So what you can see is these gray windows here are sort of out of frame, so to speak. And then I've tried to time match as best as possible at 1 month, 3 months, 6 months, 12 months, so on and so forth, between the hospitalized group, which is in red, non-hospitalized in blue, the mRNA in green, vector mRNA in purple. And essentially the summary here was, regardless of infection or vaccination, the durability is more or less the same. There's neither are superior, which was also kind of nice to see, as some people argued, the infection was better or whatever, but it doesn't seem to be the case. What was even nicer to see, though, is following vaccination, in the top here, we have the infection groups. There seemed to be a more durable response. So if you look at 6 months, for example, post 6 months after vaccination was higher than the 6 months post-infection. So what we then believe is then the additional antigen exposure increases the durability of your antibody response, which was kind of nice. Unfortunately, we couldn't get the same data for the vaccine group because a lot of them got infected. So we couldn't plot that, unfortunately, but assume more or less the same. And I'll go into more details about this particular group later. So based off these windows in the white, that's basically what we see on the right-hand side here. So the graph A is just at 1 month, and we're comparing non-hospitalized, hospitalized, mRNA, vector, all at 1 month post-infection or vaccination. And to no surprise, the hospitalized, which had a very severe infection, they had very high neutralizing antibody responses, which was the highest out of all the groups. But the vaccine groups had higher than the non-hospitalized. So essentially showing the same thing as the first paper we did, but to a more expanded level, which was kind of nice. B is at 6 months post-infection or vaccination. And again, what you see is more or less the same, but to a lower extent. So what we've kind of found was the initial antibody response kind of defines your later antibody response in terms of the level. So that you induce more to start with, you'll have higher later on as well. And then lastly, C, this is 1 month post-vaccination for the infection groups to the left. And you can see the non-hospitalized is now almost equal with the hospitalized. And then we have mRNA, 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 so 3 vaccinations. And we have vector, mRNA, mRNA. And then we also have that plus infection, so an additional antigen exposure. And the only thing I can really say here is that in the triple mRNA group plus infection, there was a nice boost. This is all against the ancestral virus, sorry. And for the vector mRNA, there wasn't a whole lot of boosting, potentially because they already had quite nice responses to begin with. So now looking at breadth, we initially had the Delta and the Omicron BA1 at the time. And the problem is trying to get all these isolates into culture takes months, and the pandemic is evolving so rapidly. So by the time we get the BA1 involved, the BA2 is here, then by the time we get the BA2, the BA5 is. So essentially we just said, okay, we're going to do the BA1 and not worry about the BA2, BA5 at the time. And so what we show here is essentially just 1 month post-infection versus 1 month post-vaccination for non-hospitalized and hospitalized. And so we have the ancestral D614G, we have the Delta, and we have the Omicron. And despite the hospitalized having such high responses initially against the Omicron BA1, they had next to nothing until they got vaccinated, and then it really boosted the titers. And you can see there's some nice responses going on there. Similarly, a bit more complicated here, we have the vaccine groups, but now we have 3 groups because we have 1 month post prime boost, 1 month post prime boost, and 1 month post prime boost infection. So there's 4 antigen exposures there. And what you can see in the mRNA group is there's an accumulation of increasing levels with each antigen exposure, as you'd expect. Not so much for the vector group, though, which is interesting. Not sure why, but up for discussion. So that was comparisons within each group. Then we obviously wanted to compare between groups. So here we have the 1 month post-infection vaccination comparison, no differences. The only interesting thing was that the vector, is my mouse coming up? Yeah, sweet. The vector mRNA group had detectable responses, whereas the other ones didn't really, with the dotted line here being the limit of detection. And then this is basically after vaccination for the hospitalized group, after prime boost for the vaccine groups, and prime boost infection for each vaccine group as well. And the only differences were against the Delta virus, really, that the hospitalized group had the most superior. That was about it. Again, so it kind of highlights here that vaccination infection is neither one's really superior. It kind of is determined individually, I'd say. So just to summarize that paper, durability of neutralizing antibody responses is similar between infection or vaccination, and the higher initial levels sort of determine your higher levels down the line as well, how much you sustain later on. Unexpectedly, antigen re-exposure boosts the durability and breadth of neutralizing antibody responses, even against, I think probably the most exciting part is you boost with an ancestral formula, but you get better Omicron responses as well. I think that was kind of nice to see. So you don't necessarily need an Omicron specific booster to get improved responses. And infection is not superior to vaccination. So now I'm going to move on to looking at a broader panel of isolates which we have now, including the more recent BQs and XBB Omicron isolates. And this is very recent work, which I just analyzed before I came back to Australia 3 weeks ago. And essentially what I wanted to look at is non-exposed vaccinated individuals. So I had 4 shots total, with the most recent shot being the bivalent Pfizer one. And then we had a sort of a hybrid immunity whereby they had 3 shots. Actually, no, sorry, right here, it's going to be 4 shots total infection at any point, and then came down to 3 shots infection vaccination to trying to simplify things and make it easier to compare. But as the N equals 28 here, hybrid basically means they had infection at any point. And when we look longitudinally at the ancestral isolate D614G, to no surprises, do we see a boost from pre-4th vaccination to post-4th vaccination for both. But the interesting thing is you can see from post-2nd vaccination to post-3rd vaccination for both groups is much higher, as you'd expect, against an ancestral formula, sorry. But post-4th, because of the bivalency, I guess, you don't see as good a boost compared to the second one. So it sort of drops off a little bit. And the other thing we kind of expected as well is pre-4th vaccination after infection for this group, they have a lot higher titers compared to the naive group. And just to reiterate, the way we classified naive versus hybrid was through the analyzer. So if they remained N antibody negative, then they were naive. We would also follow up with patient history, asking them about PCR tests and all that sort of stuff as well, to kind of confirm as best we can that they weren't exposed. So looking at breadth, it's a lot of work to do all these antibody responses, particularly with serodilution. So I didn't want to do all 28, particularly when they have infections at all different times. So the criteria was to narrow it down and match it as best as possible to the naive group, and basically look at the hybrid group post third booster when only Omicron was really around. You wouldn't really get any other variants. And so what that meant was we could kind of really simplify the data. And so looking at the naive group specifically, we have post third vaccination versus post fourth vaccination in all the different variants down here. And as you'd expect, in all the Omicrons, you get this nice boosting. But interestingly, you don't really get a boost to the ancestral isolate, even though that is part of the formula. Did the same thing in the hybrid group, but also looked at pre-fourth vaccination, because this is post-infection. And what's nice to see is you do get a boost pre-fourth vaccination, i.e. post-infection, to all the Omicron isolates, and an even further boosting after the vaccination. Which is, I guess, expected as well, but it's just nice to see. So the last thing was then to compare the two groups to make sure what we saw was correct. And so what we expected was post-third vaccination, neither group would have been exposed to any infection. And you can see there's no difference in any of the isolates. Interestingly, though, the BA2 is more neutralization sensitive compared to the BA1 or BA5. I'm not sure if anyone else sees the same results, but I thought that was quite interesting. But the newer ones are extremely resistant. When we compared post-fourth vaccination, you can see that the hybrid group is pretty much superior against every isolate, which is to be expected as well. That's not the whole study. I did a whole lot of T-cell analyses, but I didn't want to include it here because I didn't want to overcomplicate anything, and kind of focus on neutralizing antibody response, which is nice and easy to follow, I think. In summary, the bivalent vaccination boosts neutralizing antibody levels. What we see is limited against the ancestral, probably not a big thing because we don't really care about that anymore as the Omicron variants are around. And I don't think we'll see a comeback of the old one. And we see a nice boosting to all the Omicron variants, indicating that we'll probably be more protected against them as well. And similarly to the last paper, additional antigen exposure through infection improves boosting in breadth. So to finish up, this is the last study I'll present, which we're currently submitting to PNAS. This is a study looking at recombinants. So we take the ancestral isolate and we only mutate the spike. So that way we have a backbone, which is completely the same all the time. We're looking only at spike mutations or substitutions. And so we developed this whole panel of these recombinants based off all the variants that have been around. And this is just a really nice figure from our world and data that shows the sort of progression of the variants ranging from the ancestral to now this Omicron XBB 1.5 and the rapidly changing evolution of the pandemic. So this is a schematic I made basically trying to highlight what each variant has. And there's nothing particularly of importance here, except that the red ones are the RBD, and that's kind of where we focused based on literature, because we know the RBD is a strong driver for neutralization resistance. And essentially, we have a lot of deletions up here as an insertion into BA1. We went up to BA275, and we've now expanded the panel to BQ 1.1 and XBB 1.5, which are the common circulating ones now as well. That's data I have to look at as soon as I get back. So in kind of a whole, we looked at all these recombinants, again, highlighting that we just looked at the spike protein. So the N protein, the E protein, everything else was the same. And essentially what we showed initially was the spike protein is the neutralizing antibody target. There was previously some thoughts that the N protein could induce neutralizing antibodies as well, but based on this data, I don't think that's the case. It's very spike driven, as we'd expect as well. We took a panel of 12 non-hospitalized individuals with ranging neutralization. So here we have the ancestral isolate, we have the ID50 values in there, and the bluer they are, the stronger the response, essentially. So we had 3 which had quite nice responses, 6 with sort of a mid-range, and 3 with extra low responses. And we also took 6 mRNA samples. These are all 1 month post-infection or prime boost vaccination. So quite early on, because we started the study so early. And the problem with that is with the evolving pandemic, these samples become obsolete because there's so much change now as well. But essentially we picked these samples because they were staff and we had access to plasma on tap if we really wanted to. So it was kind of nice. On the x-axis down here, we have basically the whole spike for that particular variant and then the RBD only. And so what we see is with the whole spike, that's the most neutralizing resistant versus the RBD only. It's a lot less neutralizing resistant, but still neutralizing resistant. And what you notice is for the BA1, BA275 and the BA5, there's almost no neutralization really detected at all, suggesting that these spikes are the driver of neutralizing resistance. And you can see, for example, for this subject, 29, that they were borderline not neutralizing anything as well at the lower level. So not introducing that neutralizing response probably means they were less protected as well, just by speculation. So just to go through everything quickly here, we have the ID50 on the y-axis, we have individual substitutions on the x-axis, and they're sort of ranked from most sensitive to most resistant. We have NH meaning non-hospitalized, so infected, mRNA meaning the vaccinated, and the dotted lines refer to the color of their limited detection. So the minimum dilution we kind of tested. It looks a bit complicated, but that's the best way to explain it. And with the lower ones, obviously at 10, anything below 10 is really a bit iffy because you can get all sorts of bits and pieces that interrupt with the virus infectivity. In any case, what we found was the main driver, as you'd expect, is 484. It's been highly reported this position, once mutated, substituted, can cause resistance. What we later found is actually it's quite specific at which substitution causes the most resistance. So for example, this 484D, which we found in cell culture, has also been reported to increase infectivity in broad cell lines as well. Not so resistant, but this A, Q, and K. Q and K found in gamma, beta, and some of the other variants of interest as well. High levels of neutralizing resistance just at this one position, indicating this is quite a strong driver for resistance. And there was a couple of other ones as well that popped up, but didn't really seem to stick in the populations of the variants as well. So what we wanted to look at next was 484 with other substitutions or deletions in this case. So taking the 484A, which was found in the Omicron BA1, and looking at, well, firstly, what happens if we take the spike and revert the 484 specifically back to its original E? And honestly, nothing really changed. It was still quite neutralizing resistant. So it's not that single mutation alone that drives the resistance. There's obviously a combination going on. And then interestingly, if we added these deletions, which are found in some of the Omicron variants or insertion here, it sort of reverts it back to a more sensitive state as well. So again, indicating that combinations are needed to sort of lock in that resistance. Over here, we have Delta specific. So we had Delta spike and RBD, introducing specific mutations as well along the way. So what I'll point out is actually when you introduce the 484A with the RBD, there's no change compared to the Delta RBD normally. When you introduce the K or the Q, then you see an increased level of neutralizing resistance. And the Delta RBD, I think, only has 2 substitutions in the RBD as well. So indicating that those 2 substitutions with these 484 are likely strong drivers for resistance as well. I don't think a whole lot of variants popped up Delta specific with this 484, but we did see them, I think, in the Indian region. I can't remember exactly, but. And again, I have to apologize because this study started quite early. So we have Alpha, which is very outdated now, but we're going to obviously publish it on anyway because we have the data. When we introduce the 484K specifically into the alpha RBD or spike, you see increases levels of neutralizing resistance. But the alpha variant only has 1 mutation in its RBD, which is 501, I believe. But then again, indicating that they're complementary. So we're trying to find these complementary sort of substitutions that work together to increase resistance and sort of inform how the spike sort of develops as well later on. Lastly, we have several variants. These are variants of interest, I believe, and there's a couple of others introduced here. I'm not entirely sure why. They were the virologists that designed this panel as well. But essentially, 484K is introducing these variants of interest, and actually the 484K in this context wasn't as neutralizing resistant as compared to the 484K by itself. But interestingly, in the Kappa RBD, when you introduce the 484Q, it stayed resistant. Again, indicating that it's a good combination for resistance there. This is the very last slide on this, because I understand substitutions are pretty boring. I'm not a strong virologist, I'm more of an immunologist, but none of these numbers really mean anything except for looking at which specific ones are important for resistance. And these are just single substitutions that we looked at within the RBD, which maybe confer resistance or sensitivity. And the only one really was significant was this V367F, which is right at the front of the RBD. So to summarize, newer variants, highly resistant to the D614G related neutralizing antibody responses. So if you're infected with the ancestral or vaccinated only with the ancestral, those neutralizing antibody responses are obviously not so great against the newer Omicron variants. The RBD is likely the main contributor to these neutralizing resistance, with particular focus on position 484. And more to reiterate that these combinations are required to really maintain that resistance or drive it back to a sensitive state. What we'd like to do as well is confirm this by taking this spike, for example, from BA1, but then introducing the RBD from the ancestral to confirm that the RBD is then the main driver. Because what we should see is that should go back to a more sensitive state with the ancestral RBD, you would expect, but we haven't done that yet. Overall summary, neutralizing antibodies are important, and they become more durable with antigen exposures. Breadth is also increased with antigen exposures. Neither infection nor vaccination are superior to each other. They're both beneficial. And the neutralizing antibody response is almost exclusively spike-specific, I would say. Although you develop antibodies to the N protein, I wouldn't say they're particularly neutralizing based on the data we just saw. And substitutions in the RBD contribute a large portion to the neutralizing resistance with particular focus on 484, as I mentioned. And what I just mentioned as well, the newer variants are highly resistant, as we know. So with that, I would like to thank my supervisor, Jens, the clinical team, particularly Nina. All these people helped me with the neutralizing assays, virologists who designed this whole panel of recombinants. Blood processing team is important, my PhD student, and of course the funding bodies. And thank you all for listening. Now, if anyone wants to learn Danish, if you can see up here. So phonetically, you'd probably pronounce the Vidovary. It's pronounced Vidor. That's how hard Danish is to learn.
Thanks, Alex. So from the first part of the talk, it sounds to me like you just might have some data to analyze for protection. Yeah, so to look at the relationship at an individual level of neutralizing seizure or breadth and the ultimate becoming infected or not?
Yes, that is also on the cards. The thing is, for example, that's only really in the non-hospitalized. Actually, you could look at the vaccine group as well. It's how to go about that is the tricky part, and specifically what time point to look at. And we do have a couple more time points we could introduce. The problem is it's just a whole lot of work for some of these new assays. It just takes a lot of time, particularly for ID50s, as opposed to like a single dilution neutralizing assay. But definitely it's on the cards to look at sort of levels of protection, I think. Because
do you have the epidemiological data? Do you know, because you really would like your population to be highly exposed and have that recorded, but not infected or actually infected. Is that data available for the cohort?
For Denmark, more so, I would say, because they're very on top of, I guess not really testing for antibodies, but initially maybe in the hospital systems. But they're very on top of the social security system, so well set up in terms of going to get your PCR tests and getting tests for N proteins or whatever. They do have good epidemiological data, particularly from Thomas Benfield, if you ever met Thomas Benfield. He's published a lot on this as well. But in terms of protection and levels which can correlate to protection, there's not a whole lot of data for that, I think. They only have like the positive versus negative antibody response, for example. It's not a level. Yeah.
I was just going to say, because of the social security numbers in Denmark, you could link that and get all the data from that from those people pretty easily. Yeah. Yeah. So that could be, good.
But that's the nice thing about Scandinavia. They have this whole social security system, which everything is linked to. So all the data is so easily available, particularly if you work in the hospital system. All the doctors have access to it, so you can get a hold of it, no worries.
Hey, thanks. It was, really nice set of data. Thank you. I was interested in the decay of the antibodies that you showed between the hospitalized and the non-hospitalized patients. So you said you didn't see any difference in the rate of decay in the hospitalized. So over time, they would still have a higher level of antibodies 6 to 12 months after vaccination. And did you test that against other variants as well, or is that just against the ancestral variant?
Just the ancestral, yeah. Again, it was picking which variant, and as soon as we got the variant into culture, then the next variant would be out, and then we get that variant into culture, and then it became.
I'm just asking because of thinking about it. Like the hospitalized patients would be more likely, I would assume, to be older.
Exactly.
And so you would have expected them to have higher, lower antibodies initially, which I think it sort of looked like they did, and then they end up in hospital, then they get higher antibodies. But I guess I'm surprised. I mean, that a year or so later, they would technically still have better protection after their. I
wouldn't say better protection in this context. So for example, from the second or third study I showed with the majority of the data, so although the hospitalized individuals had the highest titer to begin with, against the Omicron variant, it was next to, they couldn't neutralize it at all. That was the 1 month, that was their peak neutralizing titer 1 month post-infection as well.
Right, but that's because they've only had one infection.
Yeah, exactly.
Yeah, but relatively speaking, they're still maintaining.
Yeah, but they maintain for sure better.
But they still need to get boosted.
But they would still need the boost to then see that neutralization against the Omicron variant, to then be protected against the Omicron variant, I'd say. Because that infection alone.
I guess it's just the maintenance of the high antibodies over time, which maybe I wasn't expecting.
I wasn't expecting it either. I thought they would decay all to like a sort of plateau level, which is then kind of maintained slowly over time, as we see with most acute viral infections as well. It was interesting to see that the higher it was initially, the higher it sort of was later on in time as well, despite age or whatever. Yeah.
Hi, thanks, Alex. I'm not sure if you looked at it, you might have, since you looked at the T cells, but the virus-specific B cells, were there any differences in the frequencies with the, I guess, non-hospitalized and hospitalized, and then whether that evens out following vaccination? That's
what I'm looking at now. Yeah, I don't have the data on that yet. We just set up the full assay for, again, it becomes complicated because if you want to look at SARS-CoV-2 specific B cells, it's then what spike protein do you use? So now we're going to use a combination of spike proteins to really define the populations, again, over time and between the groups as well. We've kind of just established that now. And then again, when I get back, it's going to be full-blown work in the cytometry lab, which is going to be nice, but yeah, a lot of work to do. But interesting to look at for sure.
I was kind of interested in individual variation of recognition of different sites, because when you showed all the individual site mutations and the population going down, like, looked very homogeneously. And you might expect that there might be the odd person who was like very good at recognizing that variant or very weak. I guess maybe within that cluster, people were going up and down, but do you see much evidence that particular individuals are, very dependent on a particular site, or it just tends to be fairly homogeneous how it changes with variant?
For these subjects, it seemed very homogeneous. But I think in the context of being infected by different variants, or if you got vaccinated by a different variant, which is unlikely for most, and we're exploring that now, but I think from the literature, what we see is there is differences in the sort of repertoire and what you target. So for example, if you're infected by a Delta, you're going to have a slightly different repertoire compared to the ancestral virus. And then how broad that is, it seems like the ancestral seems to be more broader targeting than the Delta virus. So I would say based on that it wouldn't be so homogeneous based on your exposure, but it's the viral or antigen exposure that defines the sort of epitopes you target, I would say, in a general sense, at an individual level, sorry. It could be differences for sure, but I haven't looked at that specifically.
Because these were all ancestral exposures,
exactly.
both the vaccines and all the infections.
Yeah.
Thanks. There's a question online, which was, how do you know, like how was exposure confirmed? So how did the participants know they were exposed, even if they weren't sick?
Yes. So PCR positive, they had to be PCR positive. The ones, we did include some that got a antibody test, but the antibody test at the time was so ambiguous that we excluded them from the data anyway. So it was PCR positive, had to be the defining result as they got infected. Except for after vaccination, then it was all based on the antibody ELISA as a determinant. Yeah. So that way we could test it ourselves rather than rely on the participant to tell us. Because although they would come in and we'd give them a survey saying, you know, if you've been infected, if there's asymptomatic, we don't really know. So the ELISA was a bit more definitive in that sense. Yeah.
With the bivalent study that you looked at, you reported that I think it was in the naive group, that there weren't very strong responses against the ancestral strain. Is that a bit different to other studies, or because it seems unusual? And what is in the bivalent?
It is it's the original formula, which has a couple of substitutions, I believe. I can't remember exactly which ones. And then the BA45 is the second one. Yeah. So I think from a B cell standpoint, having a bivalent vaccine maybe can confuse the body a little bit, because you already have predefined B cell repertoire towards the ancestral. Then you suddenly introduce that, again, ancestral plus another one. I think you'd probably get predominantly, although not looking by this data, an expansion of B cells that are already there that target what they had previously. So this immune imprinting effect. And then with the BA45, it's sort of, you have to get a de novo response, or you're relying on antibodies or B cells that are cross-reactive, and then they'll expand it that way, I think. And in that sense, I think it would be better to not have a bivalent. It would be better to have a single monovalent, but a different variant. So just have BA45. Then you specifically select cross-reactive B cells to make cross-reactive antibodies then. I think, from speculation, I don't know.
Any other? I'll ask the last question. I thought it was quite interesting even though you weren't so interested as a virologist, but as a virologist, the single substitution mutations I thought were quite interesting, particularly some of the work with Delta showing that only a small number of mutations are required to make it resistant, because I think we still are seeing Delta circulating at very low levels, and it's even recombined with an Omicron to create one of those variants. So
the Deltacron or Delta.
Yeah. So I think it is, I mean, I guess sort of relaying that information in with some of those titers that you saw, do you think it's possible that we could see one of those more ancestral variants reemerge, sort of?
Potentially. I mean, anything's really possible with this pandemic, I think. Like, where the hell did the Omicron come from? It had went from 9 substitutions to 32 substitutions, something like that. So, I mean, I think as far as the virus goes, it sort of has to combat fitness and immunity, as you know. And so maybe if the immunity sort of shifts towards the Omicron state, there could be a reversion back to a more ancestral state, providing that's also, we know it's fit, but also I don't know specifically if you're exposed to Omicron, how well you neutralize the ancestral virus, for example. But then if that's the case, they could see a shift back for sure. Yeah, I would say.
All right. No more questions online? All right. Well, thank you very much, Alex, for coming and giving a presentation of your latest work. It's very impressive. So thank you.